Hello class. So this is the first part of the series of carbohydrates. And so carbohydrates have this empirical formula shown right here. And they're mostly made up of this particular formula, but some contains nitrogen, phosphorus, or sulfur. And for this particular lecture, we will be talking about three major classes of carbohydrates, which is the monosaccharides, oligosaccharides, and polysaccharides. So the monosaccharides is just the simple sugar. You can almost think of it as like a monomer. And it consists of a single polyhydroxy aldehyde or ketone unit. Then we have the oligosaccharides, which consist of a short chain of monosaccharides units that are joined together. This particular linkage is called the glycosidic bond. And then you can have a polysaccharides, which is a sugar polymer that contains more than 10 monosaccharides units, and it could even consist of hundreds or thousands of units. So going into the first class, which is the monosaccharides, they have two different families, which is the aldose or the ketose. And what differentiates these two families is the functionality. You can either have the aldehyde, which is that carbonyl with the hydrogen on one end of the carbonyl and a carbon on the other end, which makes that aldehyde, or having a ketose, which has the functionality of a ketone, where you have the carbonyl and having the carbon on each side. And so as far as with these two families, as far as the nomenclature of it all, it usually ends with a OSE at the end, as you can see right here with glucose, and then the aldohexose, and then the ketohexose, and so on and so forth. And right in front of the O's is usually tri, tetra, pentose, hexose. All that is telling the actual chain of carbon. And so hexose is 6, so that you have 1, 2, 3, 4, 5, 6 carbons. Then you have the hexose right here would have 6, and then you have pentose, which is 5 carbons, so 1, 2, 3, 4, 5. And then right in front of the naming nomenclature of the carbon chain is the designation whether or not you have an aldehyde or a ketose. And that's when you have the aldo in front, and aldo will signify an aldehyde, which you see right here. So you have an aldo, and once again 6 carbons, and this is a carbohydrate which ends with the OSE, and so that's how it has this family name of aldohexose. Then ketohexose still has 6 carbons, but now we know with this functionality you have a ketone that is involved. So you have that carbonyl and you have the carbon on each side. And then you have the aldopentose, which means you have the aldehyde, which you see right here, and a chain of 5 carbons, 1, 2, 3, 4, 5, and it is a carbohydrate, so it ends with the OSE at the end. And then just some other things to keep in mind when you have both hexose that's happening right here. You have a D-glucose and a D-fructose. And so the difference between glucose and the fructose is glucose has an aldehyde and the fructose has a ketone. But they have the same length of the carbon chain. And then as far as for pentose, the difference between a D-ribose and 2-deoxy-D-ribose is on that second carbon that's right here. You'll notice that this carbon has the hydroxyl group, where this one does not have the hydroxyl group, and that's where that naming nomenclature of the dioxy is coming from. And then some other things just to keep note as far as with ketose. Sometimes in front of it, it has this UL nomenclature that is in front of the ose, and in between telling how many carbon chain we have, and designating whether or not we have a carbohydrate. And so we also have this Fisher projection formula of these carbohydrates. And so they have an example of D-glyceroldehyde and then an L-glyceroldehyde. And so this is very similar to what we've seen with amino acids. We have this chiral center. And as far as the isomer, this right here has a D stereoisomer, where we know as far as the highest priority is this hydroxyl group. Second is what's highlighted in yellow, and then the fourth group blocks things. So now we know we need to go in a clockwise direction, and that shows that it is a D. And for the L, the highest priority is this hydroxyl group. Then it starts to go right with the highlighted yellow, but then gets blocked again. So we go the opposite direction, and that makes it a L stereoisomer. And then the difference between the D and L, if you put a line in between here, you'll see once again that you have this mirror image, which is demonstrated right here. And just know that these two are enantiomers of each other. And nearly all sugars in biology is D. And so as far as trying to figure out whether or not you have a D or L, what you want to do is, usually with this Fisher projection, what the carbon that is the most oxidized state is at the top, which is highlighted yellow, and then you make your way down, and it's projected away from you. And so the second carbon that is to the last carbon is what is dictating whether or not you have a D or an L. So if you have a D, the hydroxyl group is to the right, and if you have an L, you can think of left, the hydroxyl group will be on the left side. And here are a series of aldose that has the D stereoisomer. And so right here you see right now with this carbon that dictates whether or not it's in a D stereoisomer or L, you see that all of these carbons have the hydroxyl group that is to the right. And so that's why these are all Ds. And so they're just showing a family of 3 carbons and 4 carbons and 5 carbons and their associated names. I'm not looking for you to memorize these all, but this more so is starting to become more so familiar with these series of carbohydrates. And then you also have the ketose versions of these carbohydrates. For this one, you don't have a chiral center that's right here, so there is no D or L configurations. But when you do have a chiral center, then that hydroxyl group that's to the right, these are all D configurations. And so once again, here are some naming configurations that come along with each of these um, different carbon chain of carbohydrates. Once again, just become a little bit familiar with it. I'm not expecting all of this to be, as far as the naming nomenclature, to be memorized for each of these different types of sugars, but this more so of the general overview of naming carbohydrates. And so not only does these monosaccharides can be enantiomers of each other's, you can also have diastereomers as well, and that's when these stereoisomers are not mirror image of each other. And so we have examples that's listed right here, where you can see if you draw a line right here, these are not mirror images of each other. And then you can also have something that's called epimers, and that is where everything is exactly the same except for one chiral center. So as you can see right here, when they show the example, this carbon, everything is exactly the same. This carbon, it is the same. But right here, this is where things change between these two different carbohydrates that are compared with each other. And then right here with the aldehyde, that is exactly the same. So only one chiral center is different. And that is what is defined as an epimer. And here are some more examples of epimers, where you have carbon that's second to last has the hydroxyl group that is to the right. So all of these are D configurations. And then from there, just notate that on the second carbon between the D mannose and D glucose, that that is the only position where where there is a difference in the configuration with the carbons, where this for the second carbon, the hydroxyl group is for the right for D glucose, but for D mannose, that hydroxyl group is to the left. But for all the other carbons, everything else is exactly the same. So that is what makes them epimers of each other's. And as far as glucose and galactose, you'll see that in the carbon 4, which is highlighted in blue, that is the only difference between these two carbohydrates, where you have an epimer that is formed because of this chiral center is different, where for this one, the hydroxyl group is for the left side, and for the glucose one, the hydroxyl group is on the right side. And so these are just summarized as the most important sugars that you should really try to start memorizing, and try to start to memorize not only this name, but the drawing of it and abbreviation and so on and so forth. These are the most important ones. You'll see in these other slides some of the structures for these carbohydrates for you to memorize. And so now, as far as with sugars, you most likely are used to seeing them as a ring structure. So this ring structure happens when there is 5 carbons or more, and it forms this rapidly and stable form of rings. And what happens is, in the general aspect of looking at it, you have your aldehyde, and it reacts with an alcohol to form this hemi acetal, and it's only a hemi because you have this ether that's right here, but it's only one, and it forms a hydroxyl group for the other. But if you have an ether here and an ether there, that is when it's a full acetal and it's no longer hemi. But you can not only have this for aldehyde with the reaction, but also with a ketone as well, and that is what is called a hemiketol type of bonds. And so when you have this 5 or more carbon chain, when you have an alcohol group within that same carbohydrates with the aldehyde, now these two can have a reaction that is very similar to this alcohol and aldehyde that was with two separate compounds. But now imagine this being part of the same chain. And so now that is how a ring can actually be formed. And so when we see the structure of a ring within carbohydrates, when you see this, this actually in the aqueous solution actually opens and closes. And so this is what we call a mutarotation. And when it closes, that carbon that is on the very right end is called the monomeric carbon. And so on this monomeric carbon, you have a hydroxyl group. When it is pointed down, that is more so of a alpha anomers. And then if you have it pointed up, it is in the beta form. And so once again, when you have a carbohydrates that has a long enough chain, one of the alcohol groups can now react with the aldehyde and then starts forming these rings. And this can also happen with the ketose version of the carbohydrates to form this open and closing ring. And once again, when the hydroxyl group is at pointing down, it is the alpha anomers, and then when it is pointing up, it's in the beta form. And so when you have these rings that are forming, 6-member oxygen-containing rings are called pyranoses, which is named after the pyran ring structure, which is shown right here. And then 5-member oxygen-containing rings are called furanoses, which is named after furan ring structure. And so when you see these type of drawings, this is what is called the Hopworth projection. And so, like I mentioned before about the Fisher projection, when you see a Fisher projection, you should be able to convert that into a Hopworth projection. And by doing so, you already know right here is that aldehyde group, which is the carbon that is right here. Because remember that oxygen group takes up a hydrogen, and so it makes a hydroxyl group. And the hydroxyl group that is right here, that oxygen is where this oxygen is correlated, and so this carbon is right here, and this carbon right here is right there. And so as you go down, so this carbon right here is associated to this carbon, and so when you see the hydroxyl group is to the right, in the Hopworth projection, the hydroxyl group is pointing down. So right is down, left is up. So this next carbon, the hydroxyl group is on to the left. That is this carbon right here, so it is pointing up. And this carbon right here, the hydroxyl group is pointing right. That is this carbon right here, and the hydroxyl group is pointing down. And so same thing right here, where you have this aldehyde, which picks up that, which forms that hydroxyl group, and that's what this carbon is. This oxygen is correlated to this hydroxyl group here. This carbon right here is associated to this carbon. This carbon right here is associated with this group. And then as we move up, this carbon right here, the hydroxyl group, is pointing to the right. That is associated to this carbon. So right is down. And then this carbon is associated with this carbon. And it is pointing right again. So it is down in the Hockworth. This takes a little bit of practice of visualizing. So, of course, I'm going to give you these slides for you to kind of sit down and look at these structures and seeing how to visualize going between the Fisher projection to the Hockworth projection. So there's a thing called reducing sugar. And so we are actually going to have a lab that correlates to this, and actually seeing sugar reducing a copper from a +2 to a +1. And so what reaction that we are going to actually be doing in lab is something called the Benedict's test, where we're going to have this copper sulfate that's within this Benedict's solution, which will end up being reduced, and then it'll be reduced to this copper oxide, and you'll see that like this red precipitation. And so this will be something that's very interesting that we will see to verify that we have sugar within our sample that we will be demonstrating in the lab. And so when the sugar acts as a reducer, the sugar itself will get oxidized. And so this property is also utilized to become more so of a glucose sensor. And so, you know, that can be correlated to trying to detect diabetes. And so what happens for a colorimetric type of analysis of glucose is when you have glucose, you have an enzyme called glucose oxidase. And then what that enzyme does is it oxidizes that sugar, and then thus producing this hydrogen peroxide, which oxidizes this organic molecule, which is a dye, and it changes color underneath another enzyme called peroxidase that helps speeds up this sort of reaction, and then produces a color change, which is correlating to the presence of glucose. And so, as I mentioned, we have this hemiacetal, where there is a hydroxyl group and ether. But when you have two ethers, this is what is more so of an acetal or a ketol type of form. And this is very important because this is the basis of trying to form this glycosidic bond, so bonding different monosaccharides together. And so this is just a general demonstration. So these reaction is what is associated with a glycosidic bond. And so where you have the anomeric carbon, once again, that is the carbon that is attached to the newly formed ether, is what is considered the glycosidic bond. And so when we go into DNA and having a nucleoside. For this instance, where you have a sugar that is bonded to a base, this is bonded to the anomeric carbon and form this glycosidic bond. But between two different sugars, that is a glycosidic bond as well, with this anomeric carbon that is bonded to the fourth carbon of another sugar. And so this glycosidic bond is very stable and does not hydrolyze readily. And so, therefore, that's anomeric carbon is no longer reactive and can't actually reduce certain metals that is around or act as a reducing sugar in general. But on the far right end, the part that does not have the glycosidic bond, that is actually reactive. But if we have these long chains of polymers, monosaccharides, then that's when the sugar becomes no longer a reducing sugar. And so, as far as starting to bind monosaccharides together, when you just have two of these monosaccharides that is bounded, is called a disaccharide. And so, once again, you have the anomeric carbon that that opens up and forms an aldehyde. So this ring opens up, and now it can react with the hydroxyl group of another carbohydrates, and then form this condensation type of reaction and form this glycosidic bond, which is right here. And this is considered acetal because you have this ether right here and this ether that is right here on both of the central anomeric carbon. But once again, just repeating myself, just make sure things are locking in. Right here on the far right end, this is the hemiacetal, because you have that hydroxyl group now, and only one of the ethers that's bonded to this anomeric carbon, and it can this ring can open and close and have this mutual rotation, and actually is active and can be a reducing sugar. But when you have two glucose that are bounded together, this new sugar is called a maltose. And then if you start binding more and more, you have these illegal saccharides, and these illegal saccharides can actually attach to protein, which is called a glycoprotein. And so a protein with small illegal saccharides attached, the carbohydrate is attached via its anomeric carbon to the amino acid on the protein, and common connections are the amino acids listed here. And then about half of the mammalian proteins are glycoproteins in general. Bacteria do not glycosylate their proteins. And then carbohydrates play actually a role in protein-protein recognitions, where viral proteins are heavily glycosylated, which helps evade the immune systems. And so protein's role is to bind specific carbohydrates, which is also called lectins or selectins. And lectins are important for many biological functions, which are listed here. And then as far as continue on with these oligomers that are being bounded to proteins, these can be oxygen linked or nitrogen linked. And so they show the example of serine that is doing the oxygen link, and this one is with asparagine, which has the nitrogen link. And this is just general education. And this concludes part one of the lecture of carbohydrates.